Hey everyone, my name is Scotland, and this is my 3D model of piezo-1. Piezo-1 is a mechanosensitive ion channel protein, and it's located in the plasma membrane of various cell types, which include red blood cells and endothelial cells that line blood vessels. It plays a key role in allowing cells to detect and respond to mechanical forces, like stretching or pressure from blood flow. Mutations in piezo-1 are associated with conditions such as hereditary xerocytosis, which is a rare red blood cell disorder, and it can also have impacts on fluid balance in the body. Piezo-1, as you can see, is shaped like a trimer, which means that it's made of 3 identical subunits that work together like a mechanical switch. When the membrane stretches, such as during blood flow, piezo-1 channel opens briefly and allows ions to flow into the cell. Specifically, it allows the inward flow of calcium and sodium ions, which help regulate cell volume, osmotic balance, and cell signaling. In red blood cells, piezo-1 helps detect when cells are compressed in narrow blood vessels and ensures they can maintain proper hydration and ion content. So let's explore the specific structure we see here with piezo-1. The protein has 3 curved subunits, which give it a propeller-like shape. Each subunit contains a transmembrane domain that spans the cell membrane, and the outer edges act as mechanosensors, while the center forms the ion-conducting pore. So to get to the specific structure we see with piezo-1, we have the primary, which is a sequence of amino acids, the secondary structure, which are the alpha helices and beta sheets. We have the tertiary structure, which is the individual folding of one subunit, and then we have the quaternary structure, which is when the 3 subunits come together to form the full shape of piezo-1. So piezo-1 does have quaternary structure. For the mutation on this protein, hereditary xerocytosis, this is a gain-of-function mutation and causes piezo-1's channel to stay open too long and open too easily. For example, in the normal protein, you have valine, which is non-polar, gets replaced with a missense mutation to glutamic acid, which is now negative. And because the channel stays open longer, too many calcium and sodium ions are allowed to enter the red blood cell, which causes them to lose potassium and water, and they'll become dehydrated and they'll become too rigid. At the cellular level, red blood cells shrink and become misshapen. They're removed too early by the spleen, which causes hemolytic anemia, fatigue, and paleness. Patients can also experience gallstones. They can experience an enlarged spleen because of this, and severe cases can lead to iron overload. In tissues like the lymphatic system, piezo-1 mutations can cause fluid retention, which leads to swelling and poor lymphatic circulation. I think this model does a pretty good job of representing the general shape of piezo-1, but there definitely are some limitations we should discuss. This model is not to scale with the individual subunits or the overall protein. It's static, but piezo-1 is able to flex. The pore is able to change shape and open in response to physical stress. And this model definitely simplifies the more complicated chemistry and the chemical interactions like hydrogen bonding, disulfide bonds that stabilize this shape and allow it to fold this way. Overall, piezo-1 is a fascinating protein. It's able to convert mechanical force into ion flow through cells and helps them adapt to their physical environment. It's interesting to see with this model how single amino acid change can cause the protein to malfunction, which can lead to serious impacts, disease, red blood cell mutations, lymphatic function. Thank you all so much for watching, and I hope you learned something.